Welcome back, folks, to our final video for this unit. In this particular video, we are going to talk about the process of meiosis, which is very similar to that of mitosis. Now, again, there are a few key differences here that we're going to take a look at, but understand that these processes are very similar in how they function, but yet produce two completely different outcomes. So let's take a look deeper. So hopefully by the end of this video, you should be able to explain the importance of meiosis, understand the individual steps of meiosis, and finally evaluate the ways that genetic diversity is ensured through the process of meiosis. So when we talk a little bit about some of the key vocabulary terms for this video, we are going to start with haploid, diploid. We'll talk about autosomes versus sex chromosomes, the process of meiosis, sperm and egg, the concept of crossing over, and the law of independent assortment. Now, when we talk about meiosis, it's important to understand the concepts of chromosome numbers, and we've talked a little bit about this already in our meiosis unit. Humans have 46 chromosomes. Half of those come from mom, and half of those come from dad. So essentially what we have is 23 pairs of chromosomes. You get one set of 23 from mom and one set of 23 from dad. Now, 22 pairs of the 23 that you have are called autosomes, and they code for a variety of different traits and characteristics and proteins that are produced inside of your body. However, you have one pair of sex chromosomes, and you've probably heard that males have an X chromosome and a Y chromosome, and females have two X chromosomes. That determines the sex of the individual that is produced as a result of fertilization. Now, your body cells, all the cells inside of your body have 46 chromosomes, and because they occur in pairs, we call them diploid. Di, of course, meaning two, meaning you have two of each chromosome. Sex cells, however, such as sperm and egg, only have 23 chromosomes. So it makes sense because when you think of the process of fertilization, when sperm meets egg, you have 23 chromosomes in sperm, 23 in egg, and they unite to form a zygote with 46 chromosomes. Then mitosis takes place and the offspring begins to develop. Sex cells are also called haploid cells, meaning that they only have one of each set of chromosomes. So key things to take away here: haploid means you have one copy of each chromosome, and diploid means you have two copies of each chromosome. 23 pairs of chromosomes exist inside of each of your cells, 22 pairs are autosomes, and one pair is sex chromosomes, which determines the sex of the offspring. So in the process of meiosis, we're undergoing a little bit of a different process here. Meiosis does not make body cells, but meiosis makes sex cells, things like sperm and egg. Rather than making two cells, like at the end of mitosis, we're making 4 cells total. Rather than having be genetically identical, all 4 cells at the end of meiosis are genetically different and contain half as many chromosomes as the original body cells. And it kind of makes sense because if you look on the right-hand side here, you see similar phases. You see prophase, metaphase, anaphase, telophase, but you see it twice. So in a way, it kind of makes sense that we're making 4 cells rather than just 2, because we're essentially undergoing mitosis twice. And that's the same reason why we have half as many chromosomes. You know, at the end of mitosis, or at the beginning of mitosis, we start with 46 chromosomes, and we end with 46. So if the process were to happen twice, and division were to happen twice, you would start with 46 and end with 23. Let's dive into a little bit more detail about how we get genetic diversity in offspring. So as I mentioned before, meiosis is essentially mitosis twice. There are a few key differences. Okay, so the first key difference is during the first metaphase. Now, if you remember, in mitosis, the chromosomes line up single file along the metaphase plate. Well, during meiosis, they actually line up in pairs. They line up in homologous pairs. So 2 of the number 1 chromosomes will pair up, 2 of the number 2 chromosomes will pair up, and so on. They'll pair up into 23 pairs of chromosomes along the metaphase plate. Now, this brings up the concept of the law of independent assortment, which means that the lineup is random for each pair. So each pair, you could have, as you see on the right-hand side here, you have kind of a purple chromosome and a blue chromosome. Each of them represent a pair of one type of chromosome. The purple can line up on the right-hand side, as it is in the picture, or on the left-hand side. keeping in mind that each of those chromosomes is genetically different from one another. So this is going to result in genetic variation based upon how they line up. That's going to affect which direction they're pulled during anaphase. We call these tetrads because there are 4 individual chromatids that are located in each pair of homologous chromosomes. So keep in mind that the way that they line up has a significant impact on genetic diversity. And the term for the pairing up of homologous chromosomes is called a tetrad. So when chromosomes do form tetrads, a very small piece of chromosome can actually exchange between the pair. So as you see, when the homologous chromosomes line up, there can actually be a little bit of a crossover. And that crossover results in more genetic variation. So you see we've got a blue chromosome, a red chromosome. They're homologous pairs, so they're a pair of the same type of chromosome. There's a crossover event that happens, and as a result, we have recombinant chromatids. We have one that is completely blue, one that's blue with a little red bit of chromosome at the end, one on the right-hand side that's completely red, and one that's mostly red with a little bit of blue. This crossing over event results in more genetic variation in the offspring that are produced. So again, when we look at meiosis 1, you can see the crossing over events that happen as they line up in pairs that form tetrads along the metaphase plate. Once metaphase happens and they're lined up, in anaphase those chromosomes are pulled apart. Telophase happens just as it would in mitosis, and cytokinesis happens, producing 2 cells. But with the second phase, now it's just more like mitosis, but instead of forming 2 cells, we form 4. So if you remember from the first set of divisions in meiosis, meiosis 1, we end up producing 2 cells that have the same number of chromosomes as the original cell. Now the process happens just like mitosis. They line up single file on the metaphase plate, the chromosomes are pulled apart, and then we end up with 4 cells that are genetically different that end up having half of the number of chromosomes that we originally started with. So again, you take a look at the entire phases compared side by side here. You have mitosis at the top, where we start with interphase, where DNA replicates, prophase, metaphase, anaphase, telophase, and cytokinesis, where we end up with 2 cells that are genetically identical, that have the same number of chromosomes as when we started. In meiosis, we have essentially 2 mitosis processes. During meiosis 1, they don't line up single file. They line up in pairs on the metaphase plate called tetrads, which result in some crossing over and some genetic variation based on how they lined up due to the laws of independent assortment. Those pairs are pulled apart, and metaphase and anaphase and telophase and cytokinesis occurs. Then the process is just like mitosis. The nucleus disappears. The chromosomes then line up along the metaphase plate, are pulled apart. Telophase happens, nucleus reforms, and then what we end up with is 4 chromosomes that are genetically different from one another, that have half the number of chromosomes from when we started. So it is a little bit of a complex process. There's a lot that's involved in here. But keep in mind that meiosis is essentially mitosis twice. Metaphase 1, the chromosomes line up in pairs versus single file, and that event can result in crossing over, and the fact that they line up randomly due to the laws of independent assortment result in genetic variation in our offspring. We end up with 4 cells, each with half the number of chromosomes, and they are genetically different from one another. So hopefully you have a good idea of how meiosis works in this video and some of the key points that occur during that process. We will talk more about this in class when you come back next time. I hope you have a great day, guys, and we will talk to you later. Bye-bye.